first, I want to introduce our Miniscope technology. It's a miniature microscope developed by Kunal Ghosh, Mark Schnitzer, and Abbas Gamal. And it is used to perform calcium imaging while the animal is freely moving. Since its inception, it has had a wide development with over 1,000 systems in over 700 sites, and we reach now over 200 plus publications and multiple industry awards. So it is now a very proven technology. But I wanted to introduce today our new platform, the new EndView, which is a platform that was initially released in 2021 for the first version. And only last month did we release the second version that has a increased sensitivity, allowing for imaging in shallow as well as deep regions with single cell resolution. The EndView is also a dual color miniscope, allowing to image in both green and red channel during naturalistic behavior. And I'm going to go over some examples showing how we can layer these signals. And we also increased the data storage for longitudinal acquisition, so you can image your animals for longer periods of time. So what can you do with this EndView platform, the most versatile miniscope? You can perform single color calcium imaging, but you can also have up to 2 color, green and red, allowing to label different cells and look at different populations activity. I'm also going to show some neurotransmitter imaging where you layer both neurotransmitter release together with calcium imaging of neuronal activity, and as well as neurovascular imaging, where now you're looking at neurons activity as well as blood flow and vessel dilation. Inscopix has worked to streamline the workflow as much as possible so that these experiments, who used to be pretty daunting, are now as easy as possible. First, we inject a virus in order to express a calcium indicator, such as GCAM. We also worked on integrated lenses to provide optical access with easy docking of the miniscope. I'm putting this tail vein injection if you want to inject a different dye in case your indicator is not a viral expression. Then you do your behavior as well as your imaging. So you have simultaneous data collection of both neuronal activity as well as behavior. And we also have tools for data processing, which I'm going to go in the next slides. So Inscopix offers not only this new EndView tool, but it's one of many products that allow this full solution with integration of behavior as well as accessories and consumables that are all working together for optimal data collection. And we also have software solutions for not only desktop data processing to do motion correction, cell extraction, and event detection, but also cloud versions for advanced data processing for batch processing as well as quality control. So let's see today some examples of this new EndView. And let's start with some calcium imaging, where here we're looking at the activity in the hippocampus using our ProView integrated lenses, 1x4 millimeter, to give optical access, and a RTI for GCAM6F, which is a ready-to-image virus that is pre-diluted to give the optimal signal during your acquisition. With this setup, in this specific animal, we were able to image as much as 500 cells activated in the field of view. This is during an open field task of about 20 minutes. Now, this is a very powerful tool, but in order to show the increase in sensitivity, I want to point out that the same, So this is a powerful tool. And here we see on the analysis, and this was done with our Ideas platform, our cloud service, where you can observe as much as 500 cells using CNMFE. The same cloud platform also allows you to visualize all of the cell traces, as well as perform some quality control of this experiment. So you'll have your cell trace, you'll have the location of the cell footprint, the off-axis, and their position towards in the field of view to make sure that those of high expression and correct signal are within the center of the field of view. You have also measurements of the bleach rate, so you can really control that each experiment that you analyze for the